Okay, let's return to the example of the person in the machine who is doing a visual task. Here we see activity in the visual cortex. To get just this part of the brain showing activity, after all, you might think that isn't there the rest of the brain doing anything? Well, they're using a certain kind of technique to illustrate just those areas that are task specific. And it's called a subtraction method, and it works like this. Disregard the PET, that's for positron emission tomography, but this is the subtraction method works for both of these imaging techniques. So let's imagine here we have the subject who is awake and doing the visual task inside the scanner, and if we looked at brain activity, we would see a lot of brain activity all over the place because there's a lot going on in the brain. But the problem is a lot of that brain activity is not specifically related to the visual task. So what you want to do is somehow subtract out all that other activity that is not task specific. And the way you do that is you also measure brain activity during some kind of controlled task. Like maybe the subject is in the scanner, but they have their eyes closed. So they're not doing the visual task. They're not seeing anything. And so all that other brain activity will still be recorded, but then you take that activity and you subtract it from the activity during the task, and you're left then with only those areas of the brain that are active or that are reasonably well defined as task specific. Right. And of course you do this on multiple subjects, and so what you see in the published papers is usually some kind of a summation or the averaged scan over several subjects. So that's called the subtraction method. So you're taking the task specific scan, subtracting the control scan to isolate those brain areas that are task specific. So let's get to the question then, why is it that the back of the brain here is turning on when the subject is seeing, right? So what is it that makes this the primary visual cortex, and why is this the auditory cortex, and why is this the somatosensory cortex? And that has to do with the connections or the inputs to these areas. Here we see another map of the cortex, and back here V1 would be area 17, and these little arrows are sort of illustrating a neural pathway. So neurons in area V1 are sending axons. They're making neural connections to neurons a little further along in the visual cortex and so on, and this goes all the way down the temporal lobe, and we'll study a little bit later how that visual processing works. But this visual information ultimately will get down into the hippocampus, as we noted earlier, the hippocampal area being a convergence zone for the sensory areas. But so what is a neural pathway? Well, again, we need to consider what neurons are. They have cell bodies, they have dendrites, and then that axon, that's the output cable, so to speak, and they terminate in a little terminal. At this terminal, called the junction of two neurons, the terminal up against the dendrite of another, is called a synapse, right? And so that would be the juncture between two neurons, and these neurons are metaphorically communicating then, right? So if this one sends an electrical signal down its output cable, little chemicals will be released here to generate an electrical signal in the target cell. That would be an example of communication between neurons. How do neurons communicate? It involves the release of chemicals across this gap, the synaptic gap, and we'll study this in upcoming lessons. But back to the question of why it is that this region is visual cortex, the answer has to do with its inputs. So here we see a diagram again of the cortex, area 17, visual cortex. If we traced back the axons that are coming up into area 17, those axons can be traced back to a structure called the thalamus. So cells in the thalamus send their axons up to visual cortex, and those cells are receiving axons from neurons in the visual, optic nerve, so the visual pathway from the eye. So the reason area 17 is visual is because it's receiving visual information along that neural pathway up to area 17. Likewise, for auditory cortex, the cells in auditory cortex are ultimately getting their neural information from cells in a neural pathway that has its beginnings in the ear. And likewise, for the somatosensory cortex, those cells are getting their neural inputs from neural pathways that have their origin on the surface of our skin. So that's what's defining these maps on the cortex, is the nature of the input going to those areas. Now, what do these sensory receptors do? Well, they're transforming different types of physical stimuli into the language of the brain, which are electrical signals that are carried by these neurons. And the communication occurs where two neurons meet at a place called the synapse. Okay, so let's just take a look at a couple more examples of these MRI images, functional MRI images. Here we see the result of a study that asked the subjects to do various tasks, for example, naming a picture that they see. So it's a visual task, so they see a picture and then they have to name it, so they actually have to say what it is, right? And the scans are broken down into sort of a temporal order. First, the first thing that happens then during a scanning episode then is that the person is seeing the picture, right? And so here we see, both in the left hemisphere and then the right hemisphere, we see the back of the brain showing high activity, right? And that would be visual areas coming down into the temporal lobe there, right? So the person is seeing the picture. Let's skip down to the last sort of temporal scan. Here we see what's happening. This is the brain areas active during the articulation. So you're naming what it is you see, right? And you're also hearing yourself speak. Well, look at what's active, right? We got temporal lobe, auditory cortex, because you're hearing yourself talk, and you've got this left hemisphere. Look at this area up in here, Broca's area, and motor cortex, right? So you're saying the name of the picture. So here we see evidence of the brain is engaging the motor systems to say what the picture is, and you're hearing yourself say it, right? Now, in between, we have these areas are active sort of throughout the whole task, called the core language areas. And here we've got Wernicke's area and Broca's area, right? So these areas are, in the left hemisphere, are being activated throughout sort of the whole task. So when you see a picture, if you know what that picture is, if you could give a name to it, right, you're kind of seeing the picture. The language areas are going to be engaged to come up with the name of it, Wernicke's area and Broca's area. And of course then the Broca's area and the motor systems then are going to be engaged to say it, and then you'll hear yourself say it. Take a look at this brain. This was a study done with congenitally blind people who are reading Braille. So if you're congenitally blind, you never saw anything in your life, so it raises the question, well, what's that back part of the brain doing? What's the visual cortex doing if it has never had visual inputs? Well, take a look. So remember, when you're reading Braille, that's a somatosensory task, so the little bumps on the page are being felt by the person's finger, and as they move their finger across the bumps, they're translating that into language. We'll take a look at the brain. Here we see the back of the brain, heavy activity in the occipital cortex there. These, in a normal subject, this would be visual cortex, but these are congenitally blind people. And so, obviously, there has been sort of a rewiring of this person's brain. What normally would be visual cortex is now being used to process tactile information as they read Braille.